I'm sure you've all heard of the wonderful 1997 movie Gattaca, starring Jude Law, Uma Thurman, and Ethan Hawke. Wonderful movie, but it means something more. Gattaca, an acronym for all of the elements that make up our DNA. Now, for many, 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 many years, for almost the entirety of human history, we have been stuck with the DNA that we were given. That's no longer the case. Through modern genetic modification techniques, we can now address our DNA and cut, improve, or replace parts of it. Now, this is an amazing subject. I have been interested in it for years, ever since my dad came home with a thesis. He works as a defense contractor for the United States military, and he came home with a paper about how this new genetic modification technique, CRISPR, can be weaponized. Kind of terrifying, isn't it? But on the flip side of it, it is also an incredible tool that can be used to modify almost any strands of DNA, even within human beings. Now, I would like to present the idea of human gene editing as a moral issue. So we will begin by going over a short portion on what it is and how we do it, and then I'll discuss some of the benefits of gene editing and some of the potential downfalls that come along with it. I should have grabbed 3 notes. All right, so there are 2 main different forms of gene editing: somatic genetic modification and germline genetic modification. Somatic begins with a fully grown host, such as an adult or a child of a human, of course. They take some cells out of the host, they culture them, they replace the parts of the DNA that they want to change, and then they replace it into the host. Those cells will then replicate and modify the host. As far as germline genetic modification is concerned, it is done on embryos and other carriers of DNA before the subject is actually formed. The interesting thing about this is that somatic genetic modification doesn't necessarily follow into the next generation of the species. Germline modification, however, is permanent. Now, how do they do it nowadays? CRISPR, clustered regularly interspaced short palindromic repeats. It's a big word, I know. But the central idea of it is that bacteria have a special defense system against viruses. What it does is it intercepts the incoming DNA and takes all the parts that would be disadvantageous for the bacteria out of it. So scientists recently have created a delivery method called Cas9, or CRISPR-associated enzyme. They call them molecular scissors because Cas9 enzyme is actually what breaks down the DNA, cuts it, and replaces it if they would so wish. All of this information is courtesy of a wonderful company called CRISPR Therapeutics. All right. There we go. This is a little bit of an image that we have here that will help expand upon the subject. There are 3 main things that CRISPR can do. It can disrupt the DNA, so it invalidates strands of your DNA that may be harmful to you. It can delete it, which completely removes it and then rejoins the strand, so that doesn't exist anymore. Or it can correct or insert portions. So, say you have a genetic disease. CRISPR can find the DNA strand where the problem originates, replace it with perhaps a correct DNA strand, and completely get rid of that disease. It's pretty incredible work. Now, let's talk about the benefits of it. Gene editing is an extremely controversial subject nowadays. However, if my note cards in the correct order, which they are not, it's a very interesting subject in the fact that there are so many incredible benefits, but most of the issues we have with them are moral. For example, it may eliminate genetic predispositions to disease and deformities. For instance, a scientist named Lang Jun, I may have fucked that up, I'm sorry, created a pair of twins that are now resistant to the HIV disease through CRISPR modification. A fascinating subject. We'll touch on that a little bit more later. Genetics may allow us to cover what humanity does best. There's a protein in our cells called myostatin. Myostatin is an inhibitor to muscle growth. So By removing myostatin, or at least reducing the amounts of it, we have discovered that we can increase muscle growth in humans just by simply removing the limiting factor. This is from a study by the Associated Press in 2019. Now, specific gene editing, we can assist our bodies in battling diseases that are already there. For instance, also looking at the study, that's it, sorry, my bad. There's a study done on a patient with genetic blindness. Now, it's something that he was born with, and he slowly went blind as the years went on. Through genetic modification, they've discovered that at a cost, he could remove his blindness. He could see again through genetic modification. Also an interesting subject we will touch on briefly. Now, the downfalls. There is a huge moral problem with genetic editing, and that case is that germline genetic modification is permanent. For all future parts of that bloodline, that modification will exist, whether it was successful or not. So future generations will not have the choice in whether or not they want gene editing. It's all up to the individual who begins it. This creates more problems such as that of eugenics or selective breeding, shall we call it. And that is why the case with He Junlang was so controversial, because without support from the scientific community, he created a pair of twins through germline modification. These twins are now permanently stuck with the genetic modifications he made to them. On the second hand, genetic modification, extremely expensive. The case I mentioned before regarding the blindness, the patient would have to pay up to from $500,000 to $1,500,000 throughout the rest of his life to maintain the genetic modifications they made, because of the body's tendency to reject new DNA. Now that we've covered both the downfalls and the upsides, I'd like to open this up to discussion with all of you.